recording. So chapter 27, it's about development and heredity. So in this chapter, we're going to start looking first into the structures that leads to the formation of a human being, including at the molecular level, which are genes. So genes, if you recall, are segments of DNA that has a particular sequence. And in that sequence, you have instructions so that you can make different structures like proteins that will make enzyme channels, and then other parts of the body to make a cell, etc. And then the genes for the maternal and paternal side has these versions, which we call alleles. And we saw the other day how, or talk about the other day about how maybe you have a different eye color than your parents. But then it turns out that that could be because maybe one of your uncles or aunts or your grandparents had the same type of eyes, color that you have. So that's inheritance. And then the genes can be seen as the blueprints then to make our tissues and organs. And these genes are present in the structure that forms the human being, that forms, in the case of human development, that forms from the fusion of a sperm cell and an egg cell that we call the zygote. So the zygote, it will undergo different changes, morphological and functional changes, to become a fetus. And now heredity, it can be defined as the transmission of genetic characteristics from parents to offspring through genes, and the study of heredity and how this happens is known as genetics. So within our DNA, our genome, we have about 3.2 million nucleotides. And these form 46 they form 46 chromosomes that pair in analogs that we call the homologous chromosomes. And are basically these chromosomes, they have the same information. Now, out of the 23 pairs of chromosomes, the first 22 are called autosomes, because independently of the gender, each of us has these genes that code for similar information within these 22 pairs. So those will be the autosomes. Now the last pair are the sex chromosomes, and it can be 2X, and it can be an X and a Y. And the 2X chromosomes, they have the same length and they have the same genetic information. So these are homologous. In the case of the Y and the X, they have different size and they carry different genetic information. So they are heterologous. And then when you study these chromosomes under the microscope, you extract them from the nucleus of the cell, you stain them, you create something and you pair them up, you take a picture, you create something that's called a karyotype. And the karyotype as a whole can give you interesting information. For instance, it can tell you if you have a normal number of chromosomes, and then it can tell you if some of your chromosomes will have pieces that are missing, or maybe they have an extra piece, or maybe you have an extra chromosome, or you're lacking a chromosome. So it gives you some general information. So that's the karyotype. And also in the karyotype, you can determine the gender of the person. So now genetic disorder will be abnormalities in the genome. It can be a single nucleotide mutation, which we call typically point mutation. It can be a deletion. It can be a translocation of genes, and then it can be an extra chromosome or lack of a chromosome. So genes are located in a specific region within the chromosome, and this is called locus. You can compare the locus to the street that you live in. So basically the street has a name and your house has a number. So that's your locus. So then we have genes with locus. And then typically in these homologous chromosomes, you have two versions of a gene, the alleles, that code for the same trait. For instance, it can be for the eye color, in the case of blue eyes. In this case, you call it homozygous, or it can be coding for blue and brown eyes. This will be heterozygous. Now, if the allele or the gene is always expressed, this will be a dominant allele. If it needs to have the pair of another one that it doesn't express all the times, this will be recessive. Now, dominant alleles are conventionally written with capital letters, while recessive with lowercase letters. So, expression of a trait coded in that allele depends in several factors. Can be environmental factors, and each individual receives one allele from each parent. The genotype will be our genetic makeup. So basically, what will be the sequence of nucleotides that we have in the genome? While the phenotype will be the physical expression. So will be the height, the eye color, skin color, hair color, etc. So we have here an example for an allele for freckles. If it's dominant, will be represented with capital letter F. And someone who doesn't have freckles, which I think most of us here, I don't see anyone with freckles, we have recessive, which is lowercase f. So if an individual has two dominant alleles for freckles, you will write it down with two capital letters F. And then if someone will have a capital letter F and lowercase f, which means he has a dominant allele and a recessive allele pair, this is called heterozygous, dominant. And if the person has two F's, lowercase, will be homozygous recessive. So then you can have three variations, FF, for homozygous because it's the same. Looks like a T. So this is homozygous dominant. And then we have heterozygous dominant And then you have homozygous recessive. Now, depending on what type of phenotype this one codes for is whether it can be the expression or non-expression. So in this case, will be no freckles for this person. This one will have freckles and this one will have freckles. So, in terms of human behavior, it's always these different couples, and then in one, you already know where I'm going. If both of them want to go to movies and they're fighting for, okay, which movie we're going, no one is going to the movies because they're dominant. You can see some dominant people here. No, we won't go. And then, in the case of heterozygous dominant, will be, well, we can go to the movie that you want this time, and the next week we can go to mine. What a relief. Happy couple. And then in this one, homozygous recessive, no, you pick, no, you pick, no, you pick. They don't have decision, so they end up not going as well. So it's crazy. So then we have some examples of how to trace for the phenotypic expression of genes based on the presence of a particular allele, dominant or recessive. So we have autosomal dominant recessive inheritance. So in this case, you have interaction between this dominant or recessive, and that includes traits like freckles, dimples, astigmatism, and including a defect in our hands like having 6 fingers. That is called polydactyly. No matter the gender, we all have autosomes, right? The 22 pairs. So in autodominant recessive inheritance, no matter if it's a male or female, the person can have freckles, dimples, astigmatism, and 6 fingers. Now, there are sex-linked inheritance in which only a gender can express those alleles. And typically, that affects males more than females. And the reason for this is because females, we have 2 X chromosomes that when one is mutated, the other one compensates for that mutation. But in males, since they don't have the other X chromosome to compensate for the mutation in the X chromosome, in that case, they always express the disease. They always have the disease. So then you use a Punnett square, which is a simple tool. It's just a square, a diagram. And then you plot in the different quadrants what type of genes comes from the parents, and then you can calculate the percentage or the probabilities. So in that Punnett square, the mother's genotype is big D, dominant, little d, recessive. And in the case of the dad, it's always dominant and dominant for the sperm. So this will be for the egg cells, and this will be for the sperm cells. So then, if you combine in this quadrant, this allele with this allele from the egg and the sperm, what would you supposed to have here? DD, which would be homozygous dominant. And then, and this is for dimples. So the person will have dimples, yes or no? Yes, so the person will have dimples. What about this combination? What would be? Heterozygous, dominant. Dimples or not dimples?
Dimples.
Dimples. Why? Because you have the dominant. What about this?
Yes.
The same, right? Will be dimples. And then for this, so what would be the percentage of progeny that would have dimples? 100%. So let's change it a little bit. What about in this case? Do you think that you would change this percentage?
Is that bottom right corner recessive?
Yes, good. Now, let's do it this way. And this will be homozygous recessive, right? Then which one's changing here? Mm-hmm. And then this one stays the same?
No, that top one changes.
Mm-hmm. Oh yeah, homozygous dominant.
Is that heterozygous?
Sorry, yes, heterozygous. Now, what about the phenotype for this?
Homozygous recessive?
Yeah, it's the same as this, but what about the phenotype? No. So then you can say easily 50-50, 50% dimples, 50% no dimples. This is a simple trait. It's kind of easy, as you saw, but it's not that easy in most of the traits. You have multiple chromosomes that code for the same function or structure or trait, and then the environmental factors play a role. It's like when someone has parents, both of them, that have diabetes. Well, you're lucky because you get those genes, but you can, if you follow a good diet, you do exercise, you can repress the chances of developing diabetes at an early stage. Okay, but then you can have something that is called incomplete dominance. So in incomplete dominance, you have this heterozygous state like this one, and not always the dominant will express. That's why it's called incomplete. So, for instance, we have sickle cell anemia, in which you have this homozygous recessive, and you produce a type of hemoglobin that is called hemoglobin S that makes the erythrocytes, instead of being circular, to be like a sickle, these instruments to cut wheat. But then you can have this heterozygous dominant in which you have the sickle cell trait and makes both hemoglobin S and the healthy hemoglobin. And individuals homozygous for normal hemoglobin, they don't produce, of course, this S hemoglobin, but these persons with sickle cell trait are healthy. They don't have this problem. So yes, in this case, sometimes the heterozygous dominant will say, well, let's always express this gene, or let's always go to a movie, but the recessive will be like, I'm tired of you telling me all the time. We went 4 times already to see whatever movie, now it's my time. So that's incomplete dominance or penetrance. It's also known as incomplete penetrance. So here we have the example then of incomplete dominant trait for sickle cell anemia. So the father's genotype will be big S, little s, and same for the mother. So they are both heterozygous dominant. So then what you can expect in the progeny is that whoever has both the dominant will be having normal hemoglobin, no anemia. These two will have the trait coming from this defective gene from the recessive. And then for the one that is homozygous recessive, will have the sickle cell. They will have the anemia. Now for multiple allele traits, again, it involves the interaction between multiple alleles. And then we have the ABO group classification, which is determined by the presence or absence of AB in the erythrocytes, if you remember. And then we have this allele for coding for A blood, which is IA, and then the one for B, which is IB. And then this will be an allele for lack of either antigen, which is I. So if you have this ABO blood type in inheritance for coding for IA and IB, you will have both expressed. This is codominant, so you will have AB blood. So if one parent is A, it has A type of blood, and it transfers this to the progeny, and then the other one is B type of blood, and transfer this allele to the progeny. In this case, you will have codominance. So here we have the table showing you what different types of blood do we have. So in this case, you have blood A for these two, blood type B for these two, and then for AB is this, and you don't code for antigens with this A and B, with this I only. So this will be type O. So someone who has an allele then that is coding for O type, but with A is going to be dominant, and then for B is going to be the same. So here is again what you can see. So the father genotype will be coding for B and also for O. Mother genotype for A and then for O. So in this case, this will be combination for AB, combination for B type, combination for A type, and then O from here and here. So for sex-linked traits, so most of the times This again comes from a mutation from the X chromosome. X chromosome, if you can see, this is an scan electron micrograph taking a picture of the X and the Y chromosome magnified 10,000 times. So see how the X chromosome is bigger than the Y. And the Y has genes that determines the gender. And then the X chromosome has a lot of genes that codes for normal development of the human brain and also for coagulation factors. In terms of the number of genes, the X chromosome has 5 times more genes than the Y, and because of that reason, females will shut down one of the two randomly. This is called the bar body. And one way to see it in the karyotype is you see all the 23 autosomes paired. By the way, how do you pair them in a karyotype? So if you have a karyotype, the largest autosomes will be pair 1. The second largest will be pair 2. The third largest will be pair 3. And you keep on going like that until you reach the smallest autosomes, which will be pair 22nd. And then you have the X and the Y, or XX. So this will be female, and this will be male. And then these are homologous. We have a lot of information. We don't need twice that information. So we shut down randomly 1, and that is going to be seen as a darker chromosome than the non-shutted, and this is called the bar body. So that's how you can tell if you see an X and a bar body, that means that it's a female. Now, what if you have XXY, or what if you have X and only X? What will be the gender? So this one will be a male, although it has 2 X chromosomes. And this one will be a female, although only has 1 X chromosome. So the absence of the Y, it is what determines the gender, not the presence of 2 X chromosomes. This is known as Klinefelter's syndrome. And it's a trisomy because you have 3 chromosomes. And then in here you have a monosomy. This is called Turner syndrome. These will be tall males, very thin. They will have mental problems because they don't develop well the brain because they have 2 active X chromosomes. And in this case, for these females, they typically are infertile because their ovaries, they become scarred, so they don't produce egg cells. And many of these females die in utero. And there are certain characteristics like they have short stature, wide neck, wide chest, the nipples are apart, and they typically are 14, 15 years old. The mom takes them to the doctor, saying to the doctor, well, she has never had her period because they have problems in the ovaries, so they are infertile. And then we have examples of diseases that are carried within the X chromosome that are X-linked, like Duchenne's muscular dystrophy, hemophilia. We have 2 types. that are carried in this X chromosome, and then color blindness. So here we have an example of red-green color blindness. So this is the combination for XR with R in uppercase, which codes for normal red-green vision. So this person is going to be a female with normal color vision. And then in this case, this X cell has a mutated gene that codes for color blindness, for red-green. So this is going to be a female that is going to be a carrier. Why? Because he has the normal gene from dad, but the abnormal gene from mom. And I told you, since we have two X chromosomes, then the gene from dad, which is normal, will compensate for the abnormality that comes from the maternal side. So that's a carrier. Do not express the color blindness. And then here we will have this normal X chromosome derived from mom and the Y chromosome from dad. So this is going to be a male with normal vision. And then in this case, this is going to be a male with color blindness because it inherited the defective gene in the X chromosome coming from mom, and then the Y chromosome from dad cannot compensate because this is a chromosome that basically just determines the gender. So colorblind. So 25% of the total progeny is going to be colorblind, and out of the males, you will have 50% males that are going to be colorblind. The other one, normal color vision. Thankfully, most inherited traits are controlled by many genes, including height, skin color, eye color. This is called polygenic inheritance pattern. And then we have multifactorial that basically takes into account the environmental factors. So here we have examples of differences in height based on polygenic inheritance. Too long, but I didn't even make it to that square. Less than 5'4". My mom was saying, de lo bueno poco. She was trying. Those moms. She was trying to console me. It's okay that you're short. No, it's not okay. Anyway, so we have techniques that allow us to screen for defects. And then among them would be Down syndrome, sickle cell anemia, hemophilia, and then you can withdraw a sample of amniotic fluid that carries cells, and then you can test for abnormalities in the chromosomes. Along with also the presence of certain molecules that can presumptively tell you the chances of having specific genetic disorders. And it is recommended that a female who's pregnant over the age of 35 at the time of delivery or at the time of delivery will be tested for these abnormalities. And this is just remembering that, of course, males can also transmit these extra chromosomes, but remember that females, our eggs, they remain arrested for a long period of time, even since we're born in meiosis. And then after we resume meiosis, we have greater chances of having chromosomes that they don't disjunct, they don't separate, and then we can transmit the most common type of trisomy that we know, which is trisomy 21, which is Down syndrome. And then you can also determine metabolic disorders for early treatment and long-term complications. One of these will be phenylketonuria. And in that case, you just, in the heel of the baby, withdraw a sample of blood with a little lancet, and then you test it for genetic defects. So then this is an example of amniocentesis. With an ultrasound, you can direct this needle to take a sample of amniotic cavity. This is done early in pregnancy, between 14 and 20 weeks of pregnancy. Or you can also take a sample of chorionic villi. We will see later on what is that, and also we will see where is the amniotic cavity and the amniotic fluid. But basically you have this needle that withdraw these cells in the vagina, is introduced, and then in the uterus, and then you take this chorionic villus sampling, and then you study for the cells that you have in that sample. So the study of then the development of the different structures in a human is called developmental biology, and well, not only in a human but also in model organisms. So you can study the changes in form and function that take place during the different periods of development. So embryology specifically study the events that occurs during the prenatal period, which the prenatal period is the first 38 weeks of life in utero, and then embryology will be the first 8 weeks. We develop after we're born, of course, that's the postnatal period, and that development happens until we die. Because as we age, there are also changes in form and function that affects us. So pregnancy embraces different events. It starts from conception, which is basically the fertilization of an egg cell or an oocyte by a sperm cell. And then the growing offspring is going to be called conceptus. Gestation period, it's 280 days or 10 lunar months, each 228 days. And then in terms of weeks, we have 40 weeks of pregnancy, and includes the 2 weeks after the mother's last menstrual period, which is when the mother usually can recall. So the prenatal period, which occurs before we're born, of course, is subdivided into 3 stages. Pre-embryonic, that includes the first 2 weeks after fertilization. And in this case, the secondary oocyte undergoes meiosis II. So remember, you have these chromosomes in metaphase plate that is waiting for the fertilization by the egg cell so that you can resume meiosis II. And then the sperm cell penetrates the corona radiata and also attaches into this zona pellucida, which is the membrane that surrounds the oocyte, because we have receptors there for the sperm cell. And then you have the formation of a single nucleus containing the 22 chromosomes from a dad, 22 chromosomes from the mom, and then the new human being, initially the form is going to be called zygote. The zygote starts dividing. So you have basically the zygote that divides in half, forming 2-cell stage. The 2 cells are going to be called blastomeres. And then these 2 cells divide in half. They form 4 blastomeres, that's the 4-cell stage. This 4-cell stage divides the cells, and then you will have 8 blastomeres. 8 blastomeres will divide, will form 16, 16, 32, 32 will form 64, 64, 128, 256. So this process, it helps for forming these subunits that are called blastomeres, that each of them is going to have unique genetic information. And that unique genetic information is going to be telling, okay, you blastomeres are going to form the heart, the blood vessels. You will form the skin cells. You will form the liver, the hair. So we have in all of the blastomeres the same genome, but the genes that you turn on and off are different. And that it is carried because the information that it is transmitted, especially from the mom side. So then our liver cells have skin cells, genes, and also have heart cell genes, and has hair cell genes, and bone cell genes, but they turn them all off, and they only turn on the liver genes. This is called differentiation and determination. Did not you have the potential, guys, to be a lawyer, to be an engineer, to be a computer scientist, rather than going to nursing? Yes, you do. Because you all have a brain, you can all go to different schools, but you differentiated to wanted to become a nurse, and out of this nursing program, once you start going and working in a hospital, you might like to go, as you explore different rotations in the hospital, you might want to go into a particular field. Maybe you like surgery, you become a surgical nurse. Maybe you like anesthesiology. So within the liver cells, not all the liver cells are liver cells. Some of them are ductules to carry the bile. Some of them are the macrophages, which are the Kupffer cells. So this is subspecialization. So our neurons are part of the nervous system tissue, but we also have microglial cells. So that's differentiation. And then one step of differentiation that happens a week after fertilization, it is called a blastocyst. The blastocyst is a hollow ball that has fluid inside, and it also has a cluster of cells that are called the inner cell mass. Out of this inner cell mass, we form the 3 germ layers, which are the endoderm, mesoderm, and ectoderm. So the embryo proper comes from these 3 layers. And this one implants within the first week. Now, before blastocyst, we have a stage in which you have a solid ball of cells that forms what is called the morula. Morula stands for mulberry, and it looks like a mulberry. So a morula will look like this. And you have early and late morula. So that happens between days 3 and 4. And then the embryonic period is extending from week 3 to week 8. And this is when you start forming rudimentary organ systems, like the nervous system, the cardiovascular system, urinary system. You start forming all of that during the embryonic period. So then this is a very critical period to form normal organs. And if a female is exposed to teratogens, which will be chemical or physical agents that they can create mutations, then the embryo can have severe damage into the organs and will not live. And then the fetal period will be after week 8, from weeks 9 through 38. And since most of these rudimentary organs were formed during embryonic period, all that happens during fetal life, it is basically enlargement of these organ systems and functionality of these organ systems. So here is the different developmental stage. Pre-embryonic period. This is the structure at day 7. This is the inner cell mass. This is the cavity of the blastocyst that is called the blastocoel. And this is a series of cells that are called the trophoblast. So this is going to implant in the superior posterior part of the uterus during during day 7. And then from weeks 3 to 8, you form all the structures of the embryo, including the future eyes, the ears, the limbs, internal organs like the brain, the spinal cord, digestive system. And weeks 9 through 38, the baby doubles in size. And then you have maturation of the function of the organs without assistance of the mother. And then just as a reminder, we talked about this in the previous chapter about infertility. So infertility basically is the inability to produce pregnancy after 1 year of unprotected sex. Several causes can lead to infertility, including azoospermia, which is low sperm count, the lack of ovulation, and then genetic defects. So in males, the most common cause will be low sperm count. In females will be hormonal problems. Maybe the female doesn't produce enough LH. LH is luteinizing hormone that leads to ovulation day 14. Maybe FSH. FSH is the one that makes these primordial follicles to become primary follicles, secondary, and then maturation of these oocytes. And then diseases of the ovary, obstruction of the uterine horns, may be due to an STD. And then in someone who desires to have progeny may undergo assisted reproductive technology or ART that involves maybe taking certain hormones or fertilization in vitro, or the use of surrogate mothers. So, fertilization in vitro, which is intrauterine artificial insemination. Basically, the sperm is placed directly into the woman's uterus at ovulation. Sorry, this is not in vitro. And then we have a gamete intrafallopian transfer or gift, in which the sperm and the oocytes are placed in the women's uterine tube. Pregnancy starts in the border between the ampulla and the infundibulum in the fallopian tube. So that's gift, basically putting the sperm and the oocytes in the uterine tube. And then you have zygote intrafallopian transfer. In this case will be a transfer of a zygote into the woman's uterine tube, and in vitro fertilization or IVF, in this case the oocytes are incubated together with the sperm in test tubes, and then you allow this fertilization to happen. You select specific zygotes, and then you have the transfer of this into the female reproductive tract. Any questions about art? Okay, so then here we have the stages of postnatal development. So we have the neonatal period that goes from birth to the first month of age. Then we have infancy, which is the first 2 years of life after the first month. And then childhood, which lasts from 2 years up to puberty. And then adolescence, which lasts from typically when it starts puberty up to the age of 19, more or less. And then adulthood or maturity can be subdivided into 3 stages: young, middle age, and late adulthood. And then senescence, which will be basically when you are basically an elderly, and that can have a lot of problems, that period, that can lead to death. Of course, someone who is in any stage of life can die, for sure, but this is related more to decaying of the functionality of most of the organ systems. And then during late adulthood, that's when you start seeing more degeneration of tissues and organs, because you have less hormones present that keep you youthful, like testosterone or even estrogens, of course, in the case of females. So preparation for fertilization begins in the pre-embryonic period. So we'll be producing gametes through meiosis, and then, of course, those gametes regularly have one end or one set of 23 chromosomes, half of the number of the diploid cells. And then when you have fertilization, you have 2N, or 46 chromosomes. You haven't erased that. So here we have 46 chromosomes. Here we have 46 chromosomes. This one will be 47 chromosomes. And this one will be 45 chromosomes. Gender. You will be XY, XX. This will be X, XY, and then XO, or Turners. So in ejaculation for a male that doesn't have azoospermia or low count of sperm cells, you have between 40 to 750 million sperm cells. And then females, we typically ovulate 1 egg cell per month. Sometimes, rarely, it can happen, but it can be 2, but typically it's 1. So then, of course, the egg cells are outnumbered by the sperm cells, and only a fraction reach the secondary oocyte, because the female has acidic secretions that can prevent the movement of these sperm cells. And then the sperm cells, besides undergoing this chemical barrier, they have to travel through the cervical canal, in which the female will have mucus. Now, if this effort to fertilize the egg happens, of course, around day 14 of the female menstrual cycle, and she has regular periods of menstrual cycle, then the mucus is going to be very thin, so it's going to allow the penetration of most sperm cells. And then the female uterus also has immune cells that can damage these sperm cells. Now, well, sperm cells go through the vagina into the cervical canal and then into the uterus. And now it has a dilemma. Should it go to the left, to the right, to the left, to the right? So it will be like that. So many of them, they end up in the race in the wrong uterine tube. So that's why out of these 40 to 750 million sperm cells, only few reach the right side and reaches the oocyte if it's in the right place and if it's viable. Now, something that happens during the first stage of the menstrual period, which is the proliferative phase, is that you have high levels of estrogen, and that makes this cervical mucus to be more watery. That helps. That promotes the sperm motility. Now, in the second phase, no matter if pregnancy happens or not, of course, with the changes in mucus, in this case, you will have a thicker mucus, then you don't expect that sperm cells will make it as easily. And then in this case, in this phase, which is called the secretory phase, you have high levels of progesterone. Does males have progesterone? Why? Why? Most of you that are on your phones, you don't even know what I'm doing. So why not? Why males, they don't have progesterone? Because they don't get pregnant. Now, in terms of viability, crazy enough, we already said that before. So, females, we have, out of our entire reproductive stage, we have more or less around 30 years of potential chances of getting pregnant, right? And we have only 12 chances within the year. And that, if you get pregnant, then you don't have a chance to get fertilized and have a baby. Males, they can reproduce anytime during the year, for many years, even after they're older, in their senescence years. But look at the life span of an oocyte, 24 hours. Out of that single day of the month, the female has 24 hours to produce progeny. Now, the sperm cells, they have a longer lifespan, around 72 hours, more or less. So, the sperm then can remain viable in the reproductive tract. It says they're up to 5 days, that's too much. So that's kind of a disadvantage in terms for us, right? But then, why I told you then that females, we have all our reproductive organs protected by these bones and the fat tissue, the mons pubis, because of that reason. Because for reproduction, we are more fragile in a way. So then ejaculation happens, and then the sperms migrate, following the track of the reproductive tract in females to try to find the secondary oocyte. And at the same time, as they're migrating, they will be trained. This is known as capacitation. So the male sperm cells, they have to be trained by the female reproductive tract so that the sperm cells will be fully motile, and then they can modify also the plasma membrane so that they can fuse into the secondary oocyte. And then the way to follow or to reach this secondary oocyte is by attraction with chemical signals, that this is known as chemotaxis. And then once they reach this secondary oocyte, the sperm cells will surround it, and then there is a lot of layers of cells that form the corona radiata, and then all the sperm cells start releasing the enzymes, the acrosomal enzymes, and they start breaking down layer by layer this corona radiata, and not as in life where the first one will be the winner. In this case, for the sperm cells, the most clever will be the winner, and it's not going to be the first one. The first ones, they will come, and then they will open up the pathway for one of the last, like to come like very lazy. You have done all the hard work, then I'm the one. I'm going to be the one that fertilizes this egg cell. So then this one that reaches the last is going to find a sperm binding receptor, a SP receptor, zona pellucida receptor, and then we'll create an acrosomal reaction. So in the acrosomal reaction, you release an enzyme that is called hyaluronidase together with acrosin, and then your plasma membrane, the zona pellucida, in the egg cell will become impermeable so that you can prevent polyspermy, fertilization of an egg cell with more than one sperm cell. So after this, the sperm will enter, and then it would shut down these sperm binding receptors. And then you will have some depolarization of these cells, and then you will have cortical granules that will cause this cortical reaction. And then additional sperm cells will be rejected. Now, something important that happens is that actually the area where the sperm cell enters into the egg cell is going to be what it will make our body to be equally symmetrical in right and left side. So that's what the sperm cells do. Basically create bilateral symmetry in us. And then once inside, the sperm nucleus will swell and form the male pronucleus, and the female will also nucleus swell and will form the female pronucleus. And then they get together, they fuse, and this is the starting point of life. This is called amphimixis. So then 23 chromosomes from paternal side, 20 chromosomes from maternal side will form the zygote. So then you will have a diploid cell. So this is the process then of fertilization. Several sperm cells come, erodes with the enzyme hyaluronidase. They have an acrosin in these acrosomal bodies in the head. They will open up gaps between the corona radiata cells. And then here we have the zona pellucida, and this is the plasma membrane of the cells. So then this one, that it is the one that enters first than the rest, will enter binding to these zona pellucida receptors, will be able to fuse and its plasma membrane and to enter. So you have the sperm cell nucleus here, it will swell, forming the pronucleus, and then this female pronucleus will also swell. And when it enters, it leaves this tail, of course, and the neck that has the mitochondria, but it brings within the centrioles. So the centrioles that we have are coming from our dad. But then the mitochondria will come from our mom and all the rest of the organelles. I mean, look at, and this is not even to scale, but look at the difference in size between the male cell and the female cell. And then the males, they only basically bring the nucleus. The rest of the cell, they leave it out, but the cytoplasm, it comes from the mom. And within that cytoplasm, guys, we have a lot of information that tells what blastomeres are going to become what. So these are what we call the morphogenetic factors that shapes us as humans. And then these two fuses form this amphimixis, this new cell, which is going to be the zygote. So this is the starting point of life. So this one contributes to 20 chromosomes, 23. So these ones that were earlier haploid now become a diploid cell. So then if someone has a mitochondrial disease, it will come from the maternal side. And then the rest will be rejected, the rest of the sperm cells. That process where the zygote divides into two cells is going to be called cleavage. And you will have a series of cleavage that are going to form the blastocyst. And these units out of this cleavage is going to be basically the formation of all the different stages between implantation and the zygote. So the zygote will split in half or cleave in half. This will be two-cell stage, undergo cleavage, you form four-cell stage. Four cells are going to form how many cells next? We have 2 more minutes. 4 will form 8, 16, so it's multiples of 2. At the 16th cell stage, you have this clump of cells that I drew there, the morula. And that's early morula because we have late morula. It looks like a mulberry. And then around day 4, this morula will be attracted into the uterine cavity because you have the endometrial glands that secretes glycogen that attracts this blastocyst or this morula because that's where food is. That's chemotaxis.